At the beautiful,
Go ahead.
repeat,
No, go ahead.
Timberlane Utility District Community Center here in Spring, Texas, a few minutes away from Mercer. We, as well as our volunteers and master naturalists and lovely guests in the audience, both in person and Zoom, are eager to learn more about a small but enchanting rare wildflower in the sunflower family, Texas Prairie Dawn. Mercer has a long history conserving Texas prairie dawn since the 1980s, after the plant, thought to be extinct, was rediscovered in Harris County. Over the last years, Mercer, along with the Coastal Prairie Conservancy and Harris County and partners in Waller, Trinity, and Gregg counties that manage preserves for populations of Texas prairie dawn, hosts a prestigious group of plant scientists from Illinois that conduct research on the biology and ecology of rare native plants, including Texas prairie dawn. Here now to share with us a little of their exciting research about the biology and ecology of Texas prairie dawn is our speaker, graduate student Julia Freeman, and with her, her major advisors, plant ecologist, Dr. Brenda Milano Flores of the University of Illinois, Urbana-Champaign, and the Illinois Natural History Survey Prairie Research Institute, and plant reproductive biologist, Dr. Janice Coons of Eastern Illinois University. Let's welcome Julia, and if you would please mute and put your questions in the chat, and we'll field those during the presentation as we're able to. So thank you so much. Here's Julia.
Hello, everybody. Thanks for bearing with us through the technical difficulties. I really appreciate it. So I'm Julia, and this is my third growing season coming out to Houston to study the Texas prairie dawn. So, every year we get to know our sites a little bit better, get to know our wonderful Texas partners a little bit better, and it's really great to be here with all of you this year. So we've got lots of time together today, so I'm happy to pause for questions as we go, and also happy to take questions at the end. So if you're here in person, feel free to raise your hand if you have a question, or if you're on Zoom, put it in the chat, and our lovely folks from the Heartwood chapter will let me know what your question is. Okay, so let's get started. Hopefully. There we go. Okay, and as Anita alluded to, none of this research happens with one person alone. So I just want to acknowledge a lot of different people who all work together to study this little rare and endangered plant species. So front and center, Anita already introduced them, but Brenda and Janice are the co-primary investigators on this project. So they are crucial to making everything happen. We also have botanist Marianne Feist, who often joins us in Texas, and Doris Lagos Coots, entomologist over at the Illinois Natural History Survey, who's been helping us with a lot of our insect work in regards to the pollinators. And then we have many wonderful undergraduate research assistants, Isabel, Maggie, and Alfred over here. So those are all the folks back in Illinois. And then over here in Texas, we have Anita, of course, here she is in action. Carrie, we didn't have a photo of you, but we appreciate you too. We have Jan Culbertson over here, hard at work counting plants, and Anna Strong with Texas Parks and Wildlife, who often joins us as well. So I'm the one who's speaking here today, but I am only a small part of this team who brings lots of expertise together to learn about these plants. And I definitely want to thank all of our Texas partners, Texas Parks and Wildlife, U.S. Fish and Wildlife Service, Mercer Arboretum and Botanical Gardens, and Coastal Prairie Conservancy, and others as well. But these are some of the big ones in Houston. Okay, so let's get started. So could you all raise your hand if you've actually been out in the field and seen the Texas prairie dawn before? Okay, a lot over that table. One here. Okay, excellent. So for those of you who have not, I want to just like set the scene a little bit. So you're out in the Gulf Coastal Prairie and it's March, so things are just kind of starting to green up. Everything's still pretty short. And you go on out to the field and you see, okay, there's this kind of low, bare brown patch. Doesn't look like much from a distance. But then you get closer and you realize, oh, that patch that looks so bare, it's not bare at all. It's filled with our little Texas prairie dawn. So all those little yellow puffballs or little yellow Q-tips out there are the blooming flower heads. And you get even closer and you get a nice good look at our little plant here. So it's got this lovely basal rosette of paddle-shaped leaves down at the ground, and then it has one or more flowering stalks. Some of them have quite a lot of flowering stalks with stem leaves. And then at the top, we have our little yellow composite flower head. So what looks to us like one yellow flower is actually made up of many tiny little yellow florets. And that's something that's important to think about when we get to the pollinator. We'll get to that later. So there we go. And some of the individuals have little white ray florets too. This one does, but they're a little hard to see. We'll have some photos with better ray florets later. So there's a little introduction. So getting more into those details. So Texas prairie dawn, its scientific name is Hymenopsis texana. It is an annual plant species, which means it completes its entire life cycle in 1 year. And it is in family Asteraceae, or the sunflower family. And it grows in these patches of salty, sandy soil that we call saline barrens. And it is endemic to eastern Texas. There are now known populations in 6 different counties, those highlighted in green there on the map. We've got that orange dot for Houston, so you'll see a lot of them are clustered around the Houston area, but then we've got a few sites further up north as well. And crucially, Texas prairie dawn is both federally and state endangered, which is one of the big reasons why we're paying so much attention to this little guy. Okay, so I've been tossing around some conservation vocab a little bit already, and so I just want to make sure we're all on the same page with some of these terms, such as rare or endangered, native, endemic. I want to make sure we all know what these mean. So a little plant conservation vocab. Rare is pretty straightforward. It's not really a technical term. It just means the species is low in abundance. There are not a lot of individuals. Federally endangered is a legal term, and in this case, we're speaking specifically about the United States. So federally endangered means any species which is in danger of extinction throughout all or a significant portion of its range. And then we have native, a species that has evolved in a particular region over a long period of time. And so we care a lot about native species because they're embedded in this web of relationships within their ecosystems. They have pollinators, they have things that eat them, they have other plants that they compete with. It's all kind of interlinked. Versus if we have a species that is not native, that is usually brought in by humans from somewhere else, it's missing all of those different ecosystem connections. And then we have endemic, which means a species that is unique to a single geographic area. So this is kind of a relative term. So let's do a little comparison. So if we take like a bur oak tree, bur oak trees are native to Texas. They have been here for a very, very long time, but they're not endemic. We have bur oak trees over in Illinois, where I live. Versus if we take Hymenopsis texana, or Texas prairie dawn, it is both native and endemic to East Texas. It is only found here. There's no Texas prairie dawn in Illinois, except for the ones in the greenhouse, and I know how they got there. So just a little comparison there. And so species that are endemic to a very small geographic area, they often tend to be more vulnerable to extinction, just because if conditions deteriorate in that one area, that's it for them. There's nowhere else where they are. And so just because a species is endemic to a small area doesn't necessarily mean that it's endangered, but it's kind of a risk factor that we can consider. And why should we care about these little yellow puffballs growing on their saline barrens? I'm guessing you all are here because you already care quite a bit about native plants. So I'm kind of preaching to the choir here. But I feel like it's always good to remind ourselves. So rare plants can have really direct benefits to us humans. For example, creating unique compounds used in life-saving drugs. I pulled just one example here. Himalayan mayapple produces the precursor to a crucial chemotherapy drug. And it is now endangered in part because of overharvesting. So we're working on creating synthetic substitutes for that drug. But anyways, shout out to the Himalayan mayapple. And then within their environments, rare plants can fill really unique ecological niches. So sometimes it's easy to think, oh, well, if it's so rare, if there aren't very many of them, then it must not matter if that plant isn't there anymore because it can't be doing very much if there's not that many of them anyways. But actually, this is often not the case. So take the example of the pitcher's thistle. It grows on sand dunes around Lake Michigan, and it blooms in July in a time period when almost no other species are blooming. And so for that little window of time, almost all the insect pollinators are coming to the pitcher's thistle. And if the pitcher's thistle is gone, then those pollinators will have nothing to eat. And then they cannot survive and pollinate all the other plants for the rest of the year. So even a rare plant can have a big impact. And then we can always just think about the existence value. This one is more philosophical, but I know many people, I'm sure many of us here believe that rare plants deserve to survive simply because they exist, and they don't need to do anything for us. Or even do anything for some bees out there in order to deserve to continue to survive and exist. Okay, so back to our prairie dawn. So why is prairie dawn federally endangered? So we talked about it being rare and it being endemic, so some risk factors there. This little screenshot is pulled from the original 1986 determination of endangered status for Texas prairie dawn. And so at the time when the proposal to list the species as endangered was first made, there were only 3 known populations of Texas prairie dawn anywhere, and 2 of them had just been destroyed by housing development. So at this point in time in 1986, there was only 1 known population left. So things felt pretty dire for the prairie dawn at that time. And as I'm sure you all are aware, living here, housing development is still a major part of the Houston and Harris County landscape and still continues to be a threat for the prairie dawn. But one of the benefits of being listed as an endangered species is it brings a lot more attention to you. And so, as I showed you that map before, we now have known populations in 6 different counties. Back in 1986, there were only populations known in Harris and Fort Bend County. And so we now have a much better understanding of how large this species range actually is. And that makes me feel a lot better and more relaxed. Not that there isn't lots of work to do in regards to conserving the Texas prairie dawn, but it's not quite as dire as we feared in 1986. And I hope that we will just keep continuing to discover more populations of these little guys. Okay, so that 1986 listing led to a 1989 recovery plan, which is now that it's listed, how do we get this species off of the endangered species list? That's actually our goal here. And so in that 1989 recovery plan, their goal was to have at least 50 distinct populations discovered, established, and protected. However, they say this may need to change since many questions about the biology and habitat requirements are still unanswered. So the 50 distinct populations, it's almost sort of a placeholder. It's their best estimate at the time, but it's really hard to say that when you don't know very much about this plant species, which we didn't. So why Brenda and Janice are out here studying the Texas prairie dawn? Well, we want to answer those unanswered questions. We want to get this species delisted. We do not want it to be an endangered species anymore. And we want to create practical guidance that we can pass on to people like Anita and Carrie, who are out there managing the Harris County Prairie Dawn Preserve. So they're on the ground, and we want to give them all the help that we can to help those prairie dawn thrive. And then finally, we are also interested in just learning more about the world, understanding more about rare plant species and their relationships with pollinators, and publishing our findings so that they can be shared with others. Okay, getting over to the research. I'll pause here. That was a lot of background. Any questions about anything I've discussed so far? Yes,
So what are the other counties north of Harris County?
yes.
And repeat the question.
Definitely. So for those on Zoom, it was what are the other counties north of Harris County? Okay, so we have Madison County over here, and then Trinity County, and then Gregg County are our three up north. Thank you. Yes.
So my question is, you've got some other counties sandwiched in there. Would it be reasonable to assume that there are some populations, they've just not been found yet?
Yeah, that's a great point. So the question was, why would it be reasonable to assume that there are other populations in these other counties that are still that are still white on the map, but are kind of in between the green counties? Yes, so folks are still out there looking. Texas Parks and Wildlife staff and U.S. Fish and Wildlife staff, they're actively keeping their eyes out for Texas prairie dawn. And that Madison County site, that one was discovered very recently, and it was just through a conversation with a landowner. So yeah, lots of potential for this species and for new populations to still be discovered. So, all right, any other questions before we move on? Okay, great. I'll have more questions at the end. Okay, so what are we actually doing out here in the field? So specifically, we are interested in the reproductive ecology of the prairie dawn. So reproductive, how does it produce? How does it make seeds and new prairie dawn plants? And then ecology is the study of organisms in their environment. So what is the relationship between an organism and all the living and non-living aspects of its environment? And so today I'm going to talk about 4 different aspects of reproductive ecology, because there's many more, but we only have so much time. So I'm going to start with a little bit about, let's see. Well, my animations aren't going, but that's fine. We're going to start with a little bit about the breeding system of the Texas prairie dawn, because that's kind of necessary background information. And then I'll spend a while with the pollinator communities, because this is what I'm focusing on for my thesis. And so it's really just where I've personally dived in. And then I also want to give you some little tidbits about all the other work that we are doing. And so I'll talk a little bit about seed dispersal and seed germination as well. All right, and the tech is being unkind to me again, so bear with me, folks.
Yeah, wait a minute.
Let's see. Let's. There we go. Oh, missed out on my pretty white boxes. Okay. Okay, hopefully I'll survive missing out on the white boxes. All right, and so now before we get into this research, I do want to put a reminder out there that all of these results are preliminary. Like I said, we are quite literally out there today, even yesterday, even gathering more data about all of these things. And then even for the data that we've gathered in past years, there's still some more statistical analyses and material processing to be done. So all the things I'm sharing with you, we're pretty sure the results really are moving in that direction, but it's not definitive yet. Okay, so starting with breeding system. So when we're talking about breeding system here, the questions that we're asking is does prairie dawn need insect pollinators at all? Many species of plant can self-pollinate and produce seeds on their own. So before we dive too deep into the pollinator communities, this is a really great question to answer. So we tried a few different things for this, and the one that ended up being most successful was a hand pollination study in a growth chamber. So this is our growth chamber over here. It kind of looks like a refrigerator, but instead of keeping food cold, it keeps plants warm and humid. And all of the bright colors you're seeing in here are various strategies to keep insects from reaching our flower heads. So we've got sticky traps, and then the flower heads themselves are also bagged with mesh bags. So ideally, me and my toothpick are the only pollinator in that growth chamber. And so, like I said, these results are still preliminary, but so far what we're learning is that prairie dawn can pollinate itself. It can produce seeds, even when no Julia with a toothpick or no insect is moving around its pollen. However, when I cross-pollinated, when I moved pollen from one plant to another plant, that increased this production of seeds. So this tells us that there likely still is an important role for insects to play in this plant system. All right, so that brings us to our pollinator community. So the first question we ask is, which insects visit prairie dawn flower heads? Now there's a little bit of a distinction here between insect visitor and pollinator. A lot of the time when we see bees or butterflies landing on flowers, we say, oh, look, they're pollinating, and that's fine. But when we're really digging into the reproductive ecology of a species, we want to be a little bit more precise. And so an insect can visit a flower head and not actually have an impact on that flower's reproduction. So what we are really looking for is not just does the insect visit, but does the insect carry pollen from plant to plant or within a flower head? And then does that mixing around of the pollen result in higher production? So it's actually kind of a pretty strict process for us to call an insect a pollinator here. I'm going to tell you a little bit about our methods. So one of our methods is 10-minute counts. So here, an individual like Brenda here observes one single little clump of flower heads for 10 minutes. So you get very complete visitation at that little clump. But of course, you can only really look at a few flower heads at a time. So you're kind of missing out on the rest of the patch. To make up for that, we also did these 30-minute walking observations. So here we are walking around a patch for 30 minutes. Any insect landing on a flower head we see, we try to either physically collect it, photograph it, or at minimum take notes on the visitor. And so here you're getting a sense of the whole patch, but not quite as in-depth. You might miss something really small.
We have a question.
Sure.
So when the plants are not cross-pollinated, do they make good seed but infertile?
Right, so they do still make fully developed reproductive seed that can germinate and become a new plant, but just not as much of it. They produce a higher percentage of infertile seeds.
Got a question over here?
Yes, thank you.
So your observer observed no fragrance protocol? In other words, they're not putting perfume on or anything like that?
That's correct, yes.
Okay.
Yeah.
That would seem to affect the.
Yeah, and that's a great point. So with all these methods, there are trade-offs, right? And so you bring up a great point that if we are there, then it's not this natural environment that it would be like if we weren't there. So it is entirely possible that our presence is disrupting the pollinator visitation. And so that's part of the reason why we use so many different methods to try to account for that. So if our presence is disruptive in some way with one method, ideally it won't be in another method. And we can put our results from all those methods together to get that more complete picture.
Or you could all wear the same perfume.
Sure. Yeah, usually not a whole lot of perfume in the field besides besides what? Yes, yeah.
I'm not sure whether the online audience can hear the question, so you might repeat it.
Yes, sorry, that question was about, are we observing no fragrance protocols in case, like, for example, if we were wearing something strongly scented, then that might affect the insect visitation. Yes, thank you. Yeah. Okay. Excellent. Okay, so our final method that I'll talk about for observing general pollinator communities, this is the one that requires no human being there at all. So we set up these Reno cameras and they take a photo every 3 seconds. And then we can put those photos together into a time-lapse video. And so this is a less disruptive way to study insect visitation. And the cameras are out there taking photos from sunrise to sunset. So it's this very complete picture, but it's focused on just a couple of flower heads at a time. So that's our limitation here. But these cameras have ended up being really helpful to us, and we've gotten some really neat footage of visiting insects from them. Okay, so let's say hello to a few of those insects we've seen interacting with the prairie dawn pollen. We are still working through the identification of these, especially some of the trickier ones. But over here we have this lovely little bee, and you can see she has some yellow Hymenaxis taxonica pollen grains on her abdomen and her leg here. Then we have this lovely little fly with some pollen grains on its hairy legs. We have this checkered skipper inserting its proboscis into the florets to get some nectar. And so we collected, I don't believe this exact checkered skipper that I have a photo of, but we collected a different checkered skipper. And I found these pollen grains down here on its little legs. Those are Hymenaxis taxonica pollen grains. Sorry about that. So these are all insects we've seen out there interacting with the prairie dawn. So I mentioned some of these. We also have this gorgeous photo of this blister beetle in family Meloidae, which we've only seen at our northernmost site up there in Gregg County. And as you can see, just absolutely stuffing its face there with the pollen. So I hope we can collect some more of those this year. So we have seen these various insect visitors engaged in pollination behavior. But the abundance has been very low. So I'm just going to give a couple examples here. In 2024, we did 33 of those 10-minute counts, 330 minutes of watching for insects, and we saw nine insects visiting the prairie dawn. In 2025, we did even more. We did 54 10-minute counts, and we saw 5 insect visitors on the prairie dawn. So we are seeing this visitation, but in very, very low numbers. However, the exception to this, low generalized insect visitation. The exception to this is the thrips, order Thysanoptera. This is a group of very tiny insects, and we have seen them on the prairie dawn in the field, in the greenhouse, and underneath the mesh that is meant to exclude insects.
Yeah.
Let's see. There we go. There's our thrips there. So we turned our attention to the thrips. So here's a little bit more about thrips. Could you all raise your hand if you've heard of thrips before? Okay. A lot of people, but not quite everybody. Excellent. So thrips are insects in order Thysanoptera. They are their own order. That means they're as distinct from other insects as beetles are distinct from moths and butterflies. And they are just very funky little critters. They have asymmetrical mandibles, so one is bigger than the other, and they use the larger one to punch into things like plant stems and pollen grains, and then they suck out the juices. And they have fringes on their wings like leather jackets, and they can inflate their feet with hemolymph, which is insect blood, and that helps them to walk straight up plants or on the underside of plants. So if you look at them under the microscope, you can see their little balloon feet. So weird. But anyway, I'm guessing you've likely heard about thrips in the context of being pests. Because they are massive agricultural and horticultural pests. And so that is where most of the research on them has focused. However, many species do feed on pollen, and they were first documented scientifically in a published scientific article as pollinators back in 1914. Since then, we have very, very, very slowly built up a body of knowledge about thrips as pollinators for more and more different plant species.
So, quick question.
Yes.
Someone saw ants on the flowers, I think, and are they pollinating?
Ants on the flowers. We have not observed ants on the flowers so far. We will be talking about ants in regards to seed dispersal, though.
Okay.
So stay tuned for that if you're an ant fan.
Okay.
Let's see. Oh, and then I just threw this in there. Once you know about thrips, you can start seeing them everywhere. This was a log I turned over in my backyard. These are fungus thrips. The adults are black and the larvae are bright pink. So that was pretty exciting. And I certainly would not have known what those were before I learned about thrips through the Texas Prairie Dawn. Okay. And so, we don't think it is all thrips that are on the prairie dawn. That's an entire order that is many, many insects. So there could be multiple species, certainly. So far, all the thrips that we've successfully identified have been Microcephalothrips abdominalis, or the composite thrips. And this makes sense because they are found in Mexico and Texas, and they often reside in those composite flower heads in the sunflower family, just like Hymenopsis texana. And they have been documented carrying pollen of a species in the same family, and they've been actually a documented pollinator in that they actually increased that production of fertile seeds for one species in the same family as the Texas prairie dawn. We're like, ooh, this is a really exciting lead. We're gonna dig more into this. Okay, so here's our questions about the thrips relationship. So firstly, how prevalent are thrips on prairie dawn? We know we've seen them, but that's kind of anecdotal. We're scientists. Let's put some numbers to this. And then we next want to ask, do thrips actually carry the prairie dawn pollen on their bodies? And then thirdly, do thrips actually increase the seed production of prairie dawn? So this third question is the most important one to us because that's what will actually tell us if we can call the thrips pollinators or not. However, it's also a very challenging question to answer well. And so you can think about these three questions as like we're trying to build a case for thrips as pollinators of prairie dawn, and we're using all the evidence that's available to us. So I'll go through these one by one. Okay, so number one, how prevalent are the thrips? So we have surveyed flower heads in the field for thrips. If any of you are going out with Jan Culbertson at the Coastal Prairie Conservancy to count the Texas prairie dawn this weekend, you too could see if you see any thrips on the flower heads. Our method is to lightly disturb the flower head by giving it a gentle squish. And then see if we see any thrips kind of come to the surface. Okay, and then we're using a second method for this. So that gives us thrips presence absence, but it doesn't tell us how many thrips are on the flower head because they are like deep buried in those florets. And so we collect a small number of prairie dawn and freeze them and then dissect the frozen ones back at the lab so we can actually count how many thrips are in the prairie dawn. And we wanted to do a comparison here because we were a little curious, do the thrips really have a special relationship with the prairie dawn, or are they just on any little yellow flower that's blooming out there? So we also collected Kriegia or dwarf dandelion, which is another small yellow flower in the same family that grows out in patches very near to the prairie dawn. And here's our results. So if you see those orange arrows there, that's pointing at 3 different thrips face down in the prairie dawn. Here's one you can see over here. So you can see they are just pointed face down into the floret. This is the position we would expect them to be in if they were actively feeding on pollen at the time when they were collected. Okay, and here's some numbers for you all. So we found thrips on about 10 to 25% of those flower heads that we squished, depending on sites. And we found a little less than 3 thrips per flower head on average. So those are our numbers. Okay, question number 2, do thrips carry pollen? So we're now taking those same thrips that were on the frozen flower heads, and now I am putting them under a microscope and counting the pollen grains stuck to their little bodies. And they do carry the pollen. So here you see we have the prairie dawn pollen grain here. It is a little lighter yellow in color and is spiky all over. And then here's the Kriegia pollen. It is a slightly deeper, more orangey yellow, and it kind of looks more like a soccer ball with just little spikes at each soccer ball point instead of being spiky all over. So they are anatomically distinct. Here's a different view with a different microscope, and you can see that those big clumps of prairie dawn pollen and Kriegia pollen on the thrips. And so on average, we found that the thrips carry about 9 grains of pollen. And I did choose some exaggerated photos there. Those are on the higher end of our spectrum. But that's important to know too, that it is possible for them to carry far more than 9 grains, even if it isn't typical. Okay, question number 3, our trickiest one. Do thrips increase seed production? So here we set up an experiment with 3 treatments. So out in the field, we have all our little flower heads in their patch. And so we are randomly assigning flower heads to one of three treatments. So either they are left open to all insect visitors, anyone can come to them, or they have a white mesh bag over them. And if you remember, back at the beginning, I showed you that photo of the thrips underneath the white mesh bag. So we know the thrips can go right through this. The white mesh bag does not keep out the thrips. We have photographic evidence. But that will keep out our larger insect visitors, our bees, our flies, our butterflies. And then we have our red mesh. So it's very difficult to exclude thrips. Thrips always want to be invited to the party. However, there's some really neat research coming out of Japan suggesting that thrips are actually deterred by the color red. We don't know why. I'm hoping someone does a study on that before I graduate, because that would be really nice. But we do have evidence that that actual color red is a deterrent to them. So we have our red mesh bags is our third treatment to physically exclude our larger flying insects and to deter the thrips. So we put those bags on when the flowers are still buds. They haven't opened at all yet, and those bags stay on throughout the entire flowering process. Then we come back and collect them when they've formed seed heads. And so we had that question earlier about what am I talking about when I'm saying seed production. And so we dissect the flower heads and we are counting up the developed fertile seeds, which are the ones with the green circle up top here. So these are nice and round and dimensional and plump. We also will find these sort of flattened, shriveled seeds that are not fully developed and will not germinate and produce flower heads. So the way we measure seed production is by seed set ratio. And so that's the number of these plump fertile seeds up top here divided by the total number of seeds. So we're working with a percentage. Okay, here we go. Here's our preliminary results for our third question. So what this is showing is our three treatments. So flower heads covered with red bags, flower heads covered with white bags, and flower heads left open to all insect visitors. And what we find is that indeed, when the thrips are deterred, the plants produce fewer fertile seeds. We have a lower percentage of fertile seeds here with the red bags, below 50%, as compared to nearly 70% for those flower heads that were left open to all insect pollinators. So we are repeating the same experiment again this year to see if we can get even more robust data on it. But so far, it's looking like the thrips actually might be more than insect visitors, which is very exciting to me.
All right, before you go on.
Yes.
We have a general pollination question, and they said they can hear me online, so you don't have to repeat this.
Oh, great. Okay.
Does cross-pollination require two different plants, or do two flowers on a single composite flower count as cross-pollination? And if on a single flower, do the genetics degrade?
That's a great question. Yes, so when we say cross-pollination here, we're referring to flowers on two different plants. It can still be helpful sometimes to have insects move pollen around within flowers on a single plant. So it's actually kind of three levels. It's whether the plant can produce seeds without pollen movement at all. It has some sort of physical mechanism that actually allows it to move its own pollen, or whether it can use its pollen from the same plant to reproduce, but it still needs to have that pollen physically moved around, usually by an insect, or does it need pollen from another plant? And let's see, what was the second part of that question?
If on a single flower, did the genetics degrade?
Sure. So very little genetic work has been done with this species. We don't actually know. All we can say is that so far our evidence suggests that when the plant only has access to its own pollen, it does not produce as many fertile seeds. So that is certainly a negative effect in and of itself.
And the last part, which I didn't read earlier, it
Yes.
says, it seems like thrips would only move the pollen across one composite head. Is that what you see?
So, we are not able to track the thrips here in Holland. And you laugh, but if we were working with bumblebees here, that is something we could try to do. Brenda and Janice have done that in the past with larger insects, but with the thrips, they're just too small. However, I will say, looking at that body of thrips pollination literature that we've slowly been building, that was one of the reasons why people didn't think thrips could be good pollinators, is that they were like, oh, we don't think they can really move around that much. But actually, we've gotten more and more evidence that they are better at directed flight than we thought initially. And so it depends on the species of thrips, it depends on the environment, but at least in some cases, the thrips actually can fly to different flower heads and different plants. So once again, we do not know how much this is happening with the prairie dawn, but it could be.
Okay.
Yes. Okay, so yes, we are building a case, and you can see these are all photos I've taken of little Microcephalothrips abdominalis on our prairie dawn. Those little black, sort of long shapes there. They're very difficult to photograph, but I'm getting better at it. Okay, so let's see. Yeah, we'll pause there. Actually, we had a few questions. Any more questions about pollinators before we move on to our next aspect of reproductive ecology?
Nope.
All right, excellent. On to seed dispersal. So the prairie dawn has produced its seeds. Now what does it do with them? How does it spread its seeds? And I want to take a moment with this picture here for you. So the seed photos that I showed you earlier were actually dissected. And so when you actually see the seeds in the seed head, they are covered in this bit of fluff called a pappus. And so this is pretty common for plants in this family, in the aster family. So if you think of a dandelion, that little bit of umbrella fluff that the dandelion has on it, that is also a pappus. So the prairie dawn has one too, it just looks a little different, not as much of an umbrella shape. Okay. So with seed dispersal, we had three potential mechanisms that we wanted to investigate. So does wind blow these seeds? Once again, kind of thinking of like a dandelion. Are they dispersed by water? Or back to the ants, that person who's curious about ants. Are the ants involved in seed dispersal? And to give you a sense of what we're looking at here, so this is a fully developed prairie dawn seed head, and you'll see that there are these stiff brown bracts that are fully closed up. So all the seeds are inside of this closed structure. So one of the things we're also wondering is like, how do the seeds get out of there? Do they just kind of grow through the seed head? Do they get out of the seed head first and then spread around? Or we're not even sure yet how that works. And so then once you crack open those bracts, this is what you see inside. You see those seeds covered with pappus. If you take off the pappus, the bare seed looks like that. And then you'll also actually see some loose pappus in there too from seeds that didn't develop at all. Okay, so first we started with wind. We did some trials in the field with these sticky traps where we're seeing if seeds are blowing around and getting stuck to the sticky traps. Once again, if you think about a dandelion, that's something that would happen, right? If you had a dandelion out there, there'd be dandelion seeds stuck to those sticky traps. We also tried it in the lab, where I was blowing that fan right on those seeds and those seed heads. And we really have found almost no evidence of the seeds being moved by wind. Even when that fan is blowing right on them, they just don't really move around. So we've kind of crossed wind off at this point. So moving on to water. So what we found is you have that dry seed head with those tough closed bracts. If you soak that seed head in water, those bracts will soften and open and expose the seeds inside. And those pappuses will kind of soak up the water and expand a little bit and sort of start nudging the other seeds out of the seed head a little bit. And this all happens pretty quickly. This can all happen in less than 10 minutes. And so if you put the seed heads in little cups of water, those bracts soften and open, this whole seed head floats and the individual seeds float as well. And so once those bracts open, you see the little seeds start to float right out of the seed head and just float around in the cup of water. And then we also tried this in the field. So I acted as the rainstorm in this case with my little watering can, and I rained on our seed heads. And then you'll see in this image, indeed, we have those little seeds coming out of the seed head, and that little seeds, the two little seeds over here dispersed out there. When the pappus gets wet, it sort of makes the star shape, which I think is really neat. So far, we've been mostly gathering evidence for water as a mechanism of seed dispersal for the prairie dawn. And this makes a lot of sense based on how they're in these low points, in these slicks that do flood very easily. You know, Anita and Carrie experienced that just last week when they were out counting the plants out at the Harris County PDP, and one of the slicks was just totally flooded, so they weren't able to count it. So this kind of makes sense thinking about that wider ecological context and the environment that the plants are in as well. And then we still needed to investigate the ants. So what did we do here? We used those Brino cameras again. So taking a photo every 3 seconds, and we set some seed heads and some loose seeds out by the red harvester ant mound, sometimes right on their little trails, like you can see here. And then we left the cameras to record what would happen to those seed heads and those seeds. Many of them stayed exactly where we left them. I think this is another argument against wind, that we can just leave some seeds out in the ground, come back a day later, and they're still there. However, some of the seed heads did not stay there. So the green circle has our little red harvester ant. The orange circle has a prairie dawn seed head, and you'll see what happens. Okay, she sees the seed head. She grabs it right away, and then she carries it. She's so strong. Look at her carrying that giant seed head. And there away she goes, kind of ducking under the grass over there, and she's off the camera frame. Okay, so me and Isabel, who used to be undergraduate, now graduate student, who works with us in the lab, we spent many hours watching these videos for the ants to come get the seed heads, and we were rewarded with sometimes these really great photos of the ants taking away the seed heads. So for seed dispersal, most of our evidence is for water dispersal and ant dispersal. And a caveat here with the ants is we have no idea how far they're taking the seeds. That would require some more in-depth experimental setups that we're not sure if we'll be able to do this time around. They could just be taking them a little bit out of the way to keep their paths clear, or they could be taking them all the way back to their nest. They could be doing something else. If they're taking them back to their nest and eating them, they would seed dispersers, for example. So it does kind of matter where the ants and the seeds end up, but it's still just really interesting to know that they will pick up and carry them. Okay, any questions about dispersal before I move on to germination?
There was one that said, have you tested wind dispersal after rain? Most rain events have wind as well.
Yes, so not in the field, just because it can be challenging for us to be out there during or right after a rainstorm. But we did do that in the lab. I tried getting the seeds wet first and then blowing the fan on them. And that did result in the most movement that we saw, which was, maybe an entire centimeter. So that is a great idea. And we did
We've lost the audio.
We often have this spring period where it will be warm, then cold, then warm, then cold, then warm, then cold. They just got a blizzard today and it was like 80 degrees a week ago. So the seeds there for many native plants have to go through this process called cold stratification, where they warm up and then they get cold and freeze again, and warm up and then they get cold and freeze again. So some people will do this in like their fridge and their freezer, and they'll be moving their seeds around. And so anyways, we Prairie Dawn probably wouldn't have had the cold stratification thing because it's here in Texas, but we didn't know if it would need something a little bit extra to get it to germinate. And so it doesn't. You just add water and it germinates. And this is true. All right, there we go. There we go. This is true even when those seeds have been in storage for a very long time. So what I'm showing you now is the germinated seeds, the percentage of germinated seeds from seeds that Mercer was keeping stored in a freezer. And the red bar, the WB Wild up top, they were collected back in 1988. And the purple and blue bars were collected in 1990. So over 35 years of storage, and we're still seeing really high germination from them. So that's very exciting. Okay, so we have learned so much about the Texas prairie dawn. They likely can self-pollinate, but it seems like seed production is improved by cross-pollination. We are seeing flower head visitation by small generalist insects and by a whole lot of thrips. And the thrips may even be pollinators of the Texas prairie dawn, but ideally we get a little bit more data on that before we claim that for real. And then we are seeing evidence for both water and ants as seed dispersal mechanisms. And then finally, we're seeing really high germination. All of this is really great and hopeful because these are all things that are kind of going well with the species. So in regards to giving people information in order to conserve this plant, possibly to reintroduce it to new areas, so far, everything's pretty easy. The thrips are out there. There's thrips everywhere. They can germinate readily. This is all great. And we still have so many questions left to go. So we want to learn more about how exactly this knowledge can be applied to things like reintroducing this species into new areas or areas where it used to be but isn't anymore. We know it's growing in this saline soil. That was not something we investigated with those germination trials. We are curious about how concentration affects the germination. Like I was mentioning, we don't really know why the ants are carrying the seed heads or how far where they're taking them. And then very little genetic work has been done on this species. So it'd be really great for some population genetics work to be done. That helps us untangle, what actually separates one population from another. Have I lost my Zoom now?
Yep.
All right. Let's see.
While you're paused, may I ask a question?
of course.
On this slide, on the germination slide, on a couple of the locations, you had a line leading up to the box, but on the others you did not. What does that line mean?
So that is showing the amount of variation in the data. So if you saw that long line out to the left, it means that we had, we have Petri dishes where we are germinating the seeds. And so, each Petri dish is going to have a different percentage of seeds that germinate. And there were some sites where we had some Petri dishes with germination that was quite lower than the average. And so that would have resulted in that long line.
Thank you.
Yeah, for sure.
You had another question while you're getting up and running there. Someone asked about the total number of seeds/flowers in the red, white, and open experiments. Did you have?
Sorry, say that again.
The total number of seeds. You talked about, you had the graph about.
With the percentage, but how
Right.
many seeds are actually on each flower head? Yes, great question. So each seed head has anywhere from 15 to about 80 seeds. So the counting process is very time intensive. There we go.
They're also asking about lookalikes. Are there plants that look enough like the prairie dawn flower that they would be misconstrued? Or are they pretty obvious, easy to identify?
Let's see. So there's another species called Hymenopsis purpygmea, or the pygmy prairie dawn. And there's some debate over whether it is fully a separate species or not. So that's definitely the one that looks the most similar. It looks so similar, we're not even sure if it's an entirely separate species. But that grows in only one area that we know of. So where you're very unlikely to encounter it. Awesome. So that's what we've been working on. And so for the last little bit about this talk, I want to kind of put it back to all of you. So what can you do to help out native or rare or endangered or endemic plants, knowing that those categories can all overlap but are not necessarily the same? So what can you do in your yard, in your community, in your city or state, or as a volunteer? So in your yard, and I don't want to assume that everyone has a yard. So when I say this could also apply to any landscaping around the office where you go to work or where you take your kids to school or a local park. But in those areas, you can actually just do less. You can have less lawn, less mowing, less cleanup of sticks and leaves and other debris that to us looks quote unquote messy, but to our insect pollinators looks like the perfect home in which to live. And you can use fewer pesticides and herbicides. This photo here is a little prairie Trillium recurvatum. Do you all have Trillium in Texas?
Yes.
No, okay, wonderful. So this lovely little spring ephemeral woodland species, it only blooms for a few weeks in the spring before the trees leaf out. And this photo I took in my yard. When I first got my home, it was all lawn. And when I stopped mowing that lawn and started killing it, this little prairie Trillium appeared. And Trillium are very difficult to grow intentionally. They take many, many years to grow. It's difficult to get the seed. And so I would never have attempted to put a Trillium on my yard, maybe for many years, but one was there. It was already there. We just had to stop mowing for it to grow. So that's very delightful. I look forward to seeing it every year. And last year it made a baby. So now there are two. Okay, and then in your community, a great thing to do is just to be a good example with native plants in your yard and talk to your neighbors about it. There's a lot of great research that is coming out about this, but I just picked out this one article to share with you today. So this was a survey of Michigan homeowners, and they found that neighborhood norms were the most important predictive factor for people preferring native plant-filled yards. So for people taking the survey, they showed them photos of different yards, some with just flat lawns, some with lots of native plants, some with like some trees and shrubs and various things in between. And homeowners in neighborhoods with ecologically innovative yards, and I think it's great phrasing, and in homeowners in neighborhoods with mixed yards, their preferred photos of a yard were those yards with 75% native plants. And they put those sort of conventional mowed lawns as last. For people who lived in neighborhoods filled with lots of conventional mowed lawns, their preferences were exactly flipped. So this can show that what you do in your neighborhood really can make a difference. And sometimes all people need to be more accepting of those natives is just to see it around them and see it as an option. And then I'll say for myself personally, I've had a really great time with my neighbor, John. When I first met John, he was kind of cranky in like a friendly way. Like he'd be like, hello, the weather's horrible today. That was kind of my relationship with John. But then John retired and he got a lot happier. He decided that his goal in retirement was to save the monarchs. And I don't think John had ever gardened before in his life. So he would just kind of throw seed packets around. He'd be like, Julia, those seeds didn't grow. And so I started giving John like divisions of perennials from my yard. And we talk about it a lot. And now he has a lot more going on in his backyard. And not all of them are natives, so I'm trying to nudge him more towards natives. So anyways, talk to your neighbors. You never know what can happen. And then in your city or state, you can be an advocate for native plants. I feel like the native plant movement is really growing. If you all have interacted with Doug Tallamy or any of his great books, I've seen lots of nods. That's wonderful. This is a really exciting movement that's going on. And so there's been some great laws passed recently in Illinois, and I was curious about Texas. And so once again, this is just one example. I'm sure there are more out there. But I saw the Texas Property Code 202.007, which has recently been updated. Which says that property owners associations may not restrict composting yard waste, rain barrels, drip irrigation, or drought-resistant landscaping, and cannot assess fines for brown grass for up to 60 days after drought restrictions. So I thought that was really hopeful and exciting. And I think the more sort of regulations we can get like that are actually like lifting restrictions, not putting more on people, but just allowing them to choose more ecological practices, the better. So I'm sure there are projects to get more of these on the books. Can join in with that. Okay, and on to the volunteers. So I'm guessing a lot of you all are here because you have volunteered with native plants in some regard. Could you raise your hands if you've done any sort of conservation or environmental or native plant volunteering? Lots of hands. All right, excellent. So you
Lots of hands up online as well.
Wonderful. What I figured, the Texas Master Naturalist and all, but I just wanted to check. So you're in very good company. 20.3% of US adults volunteer each year, and that's just with sort of defined organizations. That's not even talking about on your own. Going to conservation more specifically, according to the National Land Trust Census report, there are 61 million acres conserved by voluntary land trusts in the USA. And the Nature Conservancy put their estimated value of volunteer labor for 2023 at $5.7 million. To give another example, the Christmas bird count is a very long-running citizen or community science project where people count birds. And so this is all volunteer collected data, and there are more than 300 peer-reviewed published scientific articles based off of Christmas bird count data alone. So volunteers can do so much for conservation.
Someone wants you to go back to the slide before, that one. So I think people want to kind of get a picture of something before we move on. I think it's something that a lot of people don't know. You don't have to have a yard.
Yes, yes.
or a traditional yard, as you call it.
There's a real battle going on. A huge battle in Texas on what you're allowed to leave in your yard.
Sure.
Very good, thank you.
Do you think they're good?
I think so.
Okay.
And just for those of you online, we are going to post the recording to YouTube, so you'll be able to go back and take another look.
All right, and then finally, volunteers make it possible to track prairie dawn populations, bringing it right back around to the prairie dawn. So this is a photo from last year out at Warren Ranch, part of the Coastal Prairie Conservancy. Jan Culbertson, formerly with U.S. Fish and Wildlife Service, now Texas Master Naturalist and other organizations. She makes this event possible, as well as all the wonderful people over at Coastal Prairie Conservancy. And they'll be out there again this Thursday, Friday, and Saturday, counting those prairie dawn and making it possible to track populations through the years. So if you are interested in joining in with that, there is some informational sheets on the table over there. Yeah, Anita, do you want to say?
And someone's just posted online.
Posting the link online.
Wonderful. And Jan has posted the link in the chat for those of you
Perfect.
on Zoom.
Thank you.
Amazing. So make sure you check that out. Yes. Okay, so volunteers are wonderful, but a kind of a challenge for aligning volunteers with the goals of conservation organizations is helping people to stick around for a long time. Generally, especially with kind of more technical tasks, if volunteers keep coming back year after year and build up their own expertise, that is better because it means that more energy can be spent on actually conserving the species of interest or restoring the habitat or whatever other conservation or environmental work you're doing, as opposed to spending time, energy, and resources on recruiting and training brand new volunteers. Not that brand new volunteers are bad. Please go be a brand new volunteer if that is of interest to you. But this is just one of the things that folks who are volunteer coordinators think about. And so for my thesis, I am also doing a survey of people, not just of plants, to better understand what motivates volunteers to be committed to a volunteering effort for the long term. And so my kind of big takeaways for you all today is that rare plants do not exist in isolation. This is why the study of ecology is so important to us. In order to understand how they function and what they need, we really have to understand their interaction with the environment, whether that's with the rain, with the seed dispersal, or whether that's with the thrips as potential pollinators. And then we also need to understand the people around them. And so those communities, whether they're insects or they're humans, understanding them can really lead to better and improved and more directed conservation efforts. And then finally, you too can make a difference for native, rare, and endangered plant species, whether that's in your own yard or whether that's as a volunteer or whether that's as a policy advocate. So I will go back to that survey slide and then I'm happy to take any additional questions.
So I would encourage those online, if you have not asked your question yet, please do. I did save one, and that was just to repeat the 6 counties with prairie dawn.
Yes, let me go back to the map. All right, this is good. I can put labels on the map next time I give this presentation. There we go. Okay, so we have prairie dawn in Harris County, where the Houston dot is, in Fort Bend County, in Waller County, and then moving further up north in Madison, Trinity, and Gregg County.
Got it. Thank you.
Mhm.
The only other comments I've gotten is this is a wonderfully informative session.
Oh, good. I'm so glad. Thank you.
All right, any other questions in the audience?
You are a wonderful speaker.
Thank you very much. Appreciate it.
Yep, everybody's just saying thank you very much. So thank you very much for joining us today. And as I said, we'll do a YouTube upload and I will notify everybody that signed up and also put it out in our various communication channels. Thank you so much for joining today, everybody.